I've been studying arbuscular mycorrhizal fungi, and we've been sequencing them from single nuclei. So now, first a quick introduction of the organism, if you don't know it. So they live in symbiosis with the roots of the plants, and they go inside the cells. Well, they develop from a spore, and they go inside the cells, where they exchange nutrients, and then they will end up forming new spores that will originate new organisms. And they are key for the plant growth. But why do I even have to go to the single nuclei to get genomes? Why cannot we just sequence them? So first of all, they don't have cell walls. They just have all the nuclei inside the mycelia, and they are floating around. Also, until now, we only see this kind of reproduction where they form a big spore and they put a lot of nuclei inside. And this can lead to thinking how can they deal with the possible variations that occur in the genome, because there is no bottleneck. So after generations, we could arrive to a point where variations get accumulated, and then we would have an heterokaryotic organism with genetically different nuclei. All these in the last years have been put into doubt, and the theory now is that they have some kind of sexual reproduction, but we still haven't seen it, and we don't know yet where would it be happening. But now, how come such an important organism for plant growth, we don't know such basic things like karyosis or reproduction? So, as many of you probably know already, it's not an easy organism to study, like many fungi. Even if it's really extended around the world, it's in symbiosis with 80% of the land plants. And from those ones, at least until now, we detected a bit more than 1,000 species with ribosomal DNA sequences. And from those, only 241 species have been described using morphological features until now. The only thing that can be used to describe is the spore shape or spore color. So now, if we want to get DNA sequences to know more about them, until now the only method to grow them was using pot culture, but we already get lower the amount of species that we can use to do that. And still, it's quite a dirty method to obtain DNA, because you always have to have it with the plant, and and you always have the sand and everything. So we can use also axenic cultures, where we grow them in a plate, but still you again need the plant, and it's even more reduced the amount of species that you can grow. So all this lead to us having only one sequence on published species. But now we developed a method where we can go back to this point, to this spore point, and avoid all the culturing to obtain DNA sequences. So we start with in-bum cultures here in the US, and we did the extraction method that everyone does, using sieves and a sucrose gradient. And then individually, one by one, we pick the spores, and we can put them individually in one Eppendorf. And then we keep them there until we go to the single cell facility. So when we go to a single cell facility, we break down the spore with a pestle, and we stain the nuclei. And then a bit of how the single cell sorter works. So we get a lot of things that get stained, because the spore is really big and has also microbes inside. And everything. So what we do is there is a laser constantly pointing to the sample, and then we have two detectors, the size detector and the granularity detector, that is how the light reflects in the particle. And then we get some kind of scatter plot where we can find communities of different things, if they are more stained, bigger, or different things like that. And then after many trials, we figure out which ones of these would be our nuclei. These nuclei then, we can tell the machine to put them into single wells. So we can put one nuclei per well or multiple nuclei per well. And then we do the multiple displacement amplification, like Tim was saying until now. So it's a non-PCR-based method, and it's good because we get very long fragments and with lower frequency. And also, one key thing of this method is the more or less random coverage bias, at least more random than in PCR-based methods. And all these, well, we then sequence the reads. So we use Illumina and Oxford Nanopore. With Illumina, to take into account the coverage bias of the amplification, we sequence 24 individually amplified nuclei that we did 24 individual libraries with them with Illumina. And with Oxford Nanopore, until now, we were using pools of nuclei that also amplified. And so the assembly, obviously, it's a tricky thing. When we were trying to assemble just the reads, it was impossible because then there are such peaks of coverage. When you have 24 sets of Illumina reads, it's crazy. So the assembly was really small. And then we tried also assembling them individually, 24 individual assemblies with Mazurka, and then using a normal assembler. But then it happens that most contigs will have differences in the extremes, and then we just get a lot of redundancy, and we were getting assemblies of even 300 megabases, so much too over the expectations. So we had to generate an assembler. When you don't have the tools, you have to make the tools. So this is the work of the team of Manfred, where they build this assembler, Grapevine, that the key thing of it is this concept of dimers, that what they do is in one contig, you will have some motifs that you will decide randomly, and then you will see how the distance between the same motif in the contig. And then that will be broken down into k-mers, so it becomes a k-dimer. And then it's when you align them in pairs of contigs, and everything gets resolved over time. And you can repeat this again and again with different motifs from the beginning. Then at the end, the result, we reassembled it with a normal sequence-based assembly. And at the end, we remove the contigs lower than 500 nucleotides. We still haven't put the nanopore assemblies, so this is something that is still going on. We're still getting reads, so it's still not there yet. But just with this method, we already got a good sequence that matches our first expectations. So we have 117 megabase genome, but still we have a quite low N50, but this would be improved with the nanopore reads. And well, I haven't said it because I don't want to talk much about names, but this is the species that we've been sequenced. And just to test how the coverage was resolved if we were doing it right when we thought about sequencing 24 different nuclei, and different nuclei will give higher coverage in different areas, so we will end up covering the whole genome. We plot them in some of the longest contigs, and we can see some areas that have maybe just one set of reads that have a good coverage there, and the rest fail to cover that well there, but then in another area, a different set of reads will cover that area. So then we hope that with this method, we are covering the whole genome, more or less. And we can do this with multiple contigs. Sometimes we get crazy amounts of coverage, but more or less it's quite resolved. And then we are still processing the assembly. We are still trying to get data from it. But one of the first things that we did is just one first quick annotation of the transposable elements. And they are right now 11% of the assembly, but probably we are missing some because of collapsing. But so we have the 24,000 transposable element insertions, and we already got some interesting results with this because we found out some of the big classes of transposable elements are this mariner or hat, that they are involved in horizontal gene transfer, and they've been seen in many species of plants and how they can move from different species. So then this can lead us to think that maybe they are not only exchanging nutrients with plants, but also DNA. But we still have to continue looking into it. And but yeah, with this method now, finally we can start from one single spore and then get one genome. That until now it was impossible. And so our goal now is to then try to resolve all these three that it's until now based only in ribosomal sequences or morphology. So we have 31 isolates that 18 of them it's already sorted and amplified. 9 of them we already got the sequence back, and 1 of them that we already did nanopore sequencing with it. So we hope that soon we will have much more information to resolve this part of the tree of the fungi. And yeah, I want to thank my lab, the bioinformatics support, the collection of INBAM, the Beaver lab in Kansas that help us with the spore extraction and culturing, and the funding. Thank you.